Again, the title here is Connectivity Domain Analysis of the Default Mode Network in Mild Traumatic Brain Injury at the Acute stage. So some background on mild TBI. There's over 1 million cases per year in the US alone, and that accounts for a $55 billion direct and indirect costs per year. Up to 30% of these subjects have long-term cognitive and emotional symptoms, and while group-level functional connectivity alterations have been reported in the literature, mild TBI is highly heterogeneous, and that's a major obstacle that we have in performing combined group and individual level investigations, which are necessary. So our hypothesis with this work is that despite this heterogeneity, mild TBI patients may follow similar patterns of alterations in higher order brain functions. And the approach that we took is to use what we call the connectivity domain to extract the default mode network and do some group level and individual level comparisons between controls and mild TBI subjects. There is a paper on this recently published in NeuroImage, and this goes into a bit more details than I'll be going into, and also mention some advantages of using the connectivity domain in this type of analysis. And some of these advantages include the ability to perform both data-driven and model-based analyses. We see greater reproducibility and improved ability to compare findings across data sets. So I strongly urge you all that are interested to take a look at this paper. So in order to explain the connectivity domain, I will compare it to the time domain. So typically to extract networks in the brain, you guys are probably all very aware with this, is somewhat new to me. But we would just arrange our resting state fMRI data directly into this matrix of space against time, and then we perform an individual component analysis to extract these networks. So what we propose for the connectivity domain is, instead of time, we have these what we call seed networks, which are essentially just regions in the brain. And now, every voxel in space, we find a connectivity weight to each seed network. And then once we organize the data this way, we can again perform ICA and get our resting state networks. And we also have this matrix here, which in the time domain was hard to do any kind of analysis on because there was lots of inter-subject variability. But what we found here is that when we do it in this domain, this matrix actually becomes very useful in making comparisons to the group and individual level. So again, we first, to do this, we define seed networks, and there's some openness to this. So we can do networks based on anatomy, based on function, based on data-driven methods. Then we select a connectivity index, which we used correlation, Pearson's correlation, but you can also use things like mutual information. So there is some freedom in this type of analysis, which is good. And this is just to reiterate that when we use the connectivity domain, we are open to now using model-based methods, so not just restricted to data-driven methods like ICA in the time domain. We can create models for different groups of subjects and do comparisons on those. It's very useful and should open doors to some new types of studies. So what we did in this work, our seed networks, we chose 145 regions of interest from the Harvard-Oxford cortical and subcortical structural atlases. The connectivity index we use is a Pearson's correlation, so that those two things allow us to construct this matrix that we perform ICA on and extract the default mode network, which we did. And you can see here a comparison of the extracted default mode network from connectivity domain and compared to time domain. You can see they're pretty similar. What we did next is we had 145 seed networks. We wanted to find which ones were actually associated with the default mode network. So what we did was we had 17 controls that we used in this study. So we performed a one-sample non-parametric t-test for all of these seed networks to extract which ones were associated with the default mode network, and they're shown here. Red indicating a positive correlation, blue indicating a inverse association with the default mode network. So with all of these, we then did a group level comparison between the 17 controls and the 17 patients, and we found 8 of these regions or seed networks where we saw a significant decrease in connectivity strength with the default mode network. The most significant being the middle temporal gyrus anterior division, which is associated with attention. So what we did was then to see if we can find a correlation of this connectivity strength with that particular region in the default mode network with this attention. We got these attention function composite scores and plotted them together for our patients and we indeed found a correlation. You can see this outlier here. So this is a really strong correlation. It was good to see. And even if you take out this outlier here, we still end up with a low p-value and a significant correlation between these two things. So hopefully this could be used as a means to predict some kind of attention, problems in patients. We also, here's the subject level comparison. So that's part of this is being able to do group and subject level comparisons. So what this chart is showing is for every patient and all these seed networks that we found to be associated with the default mode network, we compare them all to the control group, and we find check marks here indicate a decreased connectivity strength with that particular seed network, and we also found we found 4 out of 60 increase connectivity strength when compared with the control subject. So in conclusion, you see that the connectivity domain helps to investigate group and individual level alterations, and despite the high heterogeneity in mild traumatic brain injury, patients share common patterns of alterations